Hi, good morning everyone. Today I will start with the introduction of our presentation. So let's see our slide. Many plant species around the world are facing threats such as habitat loss, climate change, and human activity like deforestation. Because of this, many plants are becoming endangered or extinct. Plant conservation is very important because plants play a major role in maintaining biodiversity and ecosystem balance. They also provide food, medicine, and resources for humans. However, traditional conservation methods sometimes have a limitation. Therefore, modern biotechnological tools such as molecular marker and genomic technique are increasingly used to support plant conservation. In this presentation, we will discuss how these biotechnological tools contribute to effective plant conservation.
Next is the application of molecular markers and genomic techniques in conservation of plant. A marker is a segment of DNA that can be identified at a specific location within the genome. This marker can help scientists to locate and track a particular region of DNA. So, DNA markers are widely used to study patterns of heredity, genomic variation, and evolutionary process, including natural selection. By comparing these DNA markers, researchers can understand how traits are inherited and how species evolve over time. Next, they are also important for species identification and taxonomic classification. Rather than the traditional methods that relies on physical characteristics, scientists nowadays can provide a more accurate genetic-based approach, especially for species that look similar in morphologically. This can help them to conserve the right species and help them thrive. Last but not least, DNA markers also play a crucial role in plant breeding. This allows researchers and breeders to identify and select plants that carry desirable genes so they are more resistant towards to pests and diseases. This will help in develop a stronger and healthier crop for more efficiently.
Genomic is study of a plant's complete DNA, including nuclear, plastid, and mitochondrial genomes. It helps researchers understand genetic diversity and relationships between plant species for conservation. Genomics contribute to plants conservation by first identifying species accurately using DNA, especially similar-looking plants. Second, detecting hybridization to maintain species purity. Last one, improving adaptability by selecting plants resistant to climate change, disease, and environmental stress. Next, genomic techniques. First, next-generation sequencing NGS to analyze whole genomes and genetic diversity. Second, DNA barcoding to identify species using short DNA sequence. Genomics enables better database decision to conserve plant biodiversity.
Next, we move to the impact of biotechnological tools in plant. First, biotechnological tools preserve plant genetic diversity through seed bank, gene bank, and cryopreservation. Cryopreservation at -196 degrees Celsius is used to conserve plant tissues and recalcitrant seeds that cannot be conserved conventionally. Second, they support plant conservation and restoration by using tissue culture and micropropagation that allow plants to multiply rapidly. It is important for restoring population of rare and endangered species. Third, this tool also enable conservation of rare and endangered species by using somatic embryogenesis. Somatic embryogenesis allows plants to be regenerated from somatic cell, which is useful for species with poor natural reproduction. Moreover, these tools can also be complement in both in situ and ex situ conservation strategy. And finally, biotechnological tools can help to reduce extinction risk from environmental threats by using molecular marker, genomics, and DNA barcoding to assess genetic diversity that can guide to conservation decisions.
The impact bases on the lecturer tool uses in plant conservation. First, tissue culture, micropropagation, and somatic embryogenesis play a very important role in conservation. This technique allows the rapid multiplication of endangered plant species in a controlled environment. By using small plant tissue, we can produce a large number of healthy and genetically identical plants in a short time. Next is cryopreservation. Cryopreservation is used for the long-term storage of plant tissue and recalcitrant seed. These materials are stored at extremely low temperature, usually in liquid nitrogen, which stops all metabolic activity. Another important tool is molecular marker, such as RAPD, FLP, SSR, and SNP. These markers are used to study genetic diversity in breeding level and population structure within plant species. The information obtained help scientists make better conservation decisions and manage population more efficiently. Lastly, genomic DNA barcoding and SPR technology provide advanced approach in plant conservation. DNA barcoding help in accurate species identification, genomics support conservation planning, and SPR can be used to improve plant survivability by enhancing resistance to disease or environmental stress.
In conclusion, biotechnology plays an important role in conserving plant diversity through techniques such as tissue culture and cryopreservation. Endangered plant species can be preserved for future use. Genomic tools like DNA barcoding and sequencing also help scientists accurately identify plant species and understand their genetic variation. Furthermore, modern technologies such as CRISPR contribute to improving plant resistance to disease and environmental stress. Overall, biotechnological and genomic approaches are essential to ensure effective and sustainable plant conservation in the future.